Hey everyone, today I'm going to explain you about some hematological sports. Okay, so there are some pipettes, there are some tubes, and there are some fluids. Okay, so these can be given in spotting in your examination of hematology only. If you are a paramedical student, these are the all spots which you can get. If you are a medical student, MBBS student, then these can be some of the sports. Apart from that, you may have some other lab sports like clinical lab, human lab, or some amphibian sports as well. Okay, so let's see. These are three pipettes which you can be given one by one. Fine. So there can be bulb, there can be without bulb pipettes. So you have to identify which pipette it is and tell the use of it. Okay, this way you can be asked some questions. So first you have to identify, if there is a bulb, then you have to identify the bead color. So this is red in color. Okay, then you have to see marking. So if there is 101 marking and red bead in color, that means it's RBC pipette used for total RBC count. Fine. Then the second pipette is, again you have to see the bead color. This is white color and the marking is 1 1 written here. Okay, 1 1. That means this is WBC pipette used for total leukocyte count. Fine. Don't go about this color of mouthpiece. These can be exchanged actually. Then there is a pipette which don't have any bulb but have one marking, only one marking here, and this unit is 20 microliter. Okay, this is written here, 20 microliter. 20 microliter. Fine. So this is hemoglobin pipette where we have to load 20 microliter blood for HB estimation. Fine. Then are some tubes. So this tube is basically one side blind tube and one side open, and if you see marking, there are 0 to 10 marking from upward to downward, and on opposite side there is 1 to 10 marking from downward to upward, vice versa. So this tube is called Wintrop's tube. Wintrop's tube is used for PCV or ESR estimation. That's why it have two values. Then there is one tube which is square shaped, and this is hemoglobinometer tube, which is having two marking. One is red color marking on one side and yellow color marking in gram percentage on other side for hemoglobin estimation, right? Then is a long tube which is open from both sides. If you can see, this is open from both the side, and this is Vestigrin pipette which is for ESR estimation. Okay. Then last is the Pasteur pipette, which is used to fill the blood in Wintrop's tube. Okay, in ESR estimation, same thing. Then is this is a box which can be given in your sports, and you can see it has some standard color on both the side. And in hemoglobin estimation, we put this hemoglobinometer tube inside here and compare the color. That's why this apparatus is called comparator. Obviously, you must be knowing what is this. These are anti-seras. Okay, for blood grouping we are using anti-A, anti-B, anti-D. Okay, so if this spot is given, what you have to answer is these are anti-seras and used for blood grouping practical. Then that you can be given all the stains, whatever practical you have done. If you can see, there are Hames fluid, Dark's fluid, Lessman stain. So these stains can be given same way labeled. Okay, then you have to tell the composition of this stain and function of this stain. So you must be knowing, for example, Hames fluid. Hames fluid will be used for total RBC count, and if it is used for RBC, then composition should be some material which prevent Rouleau formation, which prevent any infection and maintain isotonicity, because RBCs are osmotic fragile, right? And that's why composition is sodium sulfate, mercury chloride, and sodium chloride, right? Then is Tux fluid, which is for WBC count, right? And hence it's for WBC count. You should be knowing that there should be lysis of membrane of RBCs and WBCs. That's why glacial acetic acid and one stain to basically stain the nucleus. Okay, then is Lessman stain. So Lessman stain is used for DLC, or basically staining a thin blood smear for identification of cells, right? So Lessman stain composition will be one acidic dye, one basic dye, and one fixative. Okay, so acidic eosine dye, basic dye methylene blue dye, and one composition which is fixative is acetone free methyl alcohol. And you can be asked why acetone free, but it's not asked in spotting. Okay, so in spotting you can be asked what is composition of this fluid and use of this fluid. So in all these sports you can be asked name the apparatus or name the fluid and one, use of it. So I hope this spotting Wi-Fi is clear to you. It can be asked in your hematological practical as well, and I hope this is clear and you'll score marks in spotting because of this. Thank you so much. If you want amphibian sports as well, you can comment below. Thank you so much. Bye bye.